E. coli RP437 are introduced into a chemotaxis device where they are exposed to defined spatial concentration gradients. Cells entering the chamber encounter the midpoint of the concentration gradient. Depending on whether the signal is perceived as an attractant or repellent, bacteria rapidly move either up or down the concentration gradient.
Hi, I'm Derek Englert from the Laboratory of Molecular Systems Biotechnology in the Department of Chemical Engineering at Texas A&M University. Today we'll show you a procedure investigating bacterial chemotaxis in a microfluidic device. We use this procedure in our laboratories to study how E. coli RP437 migrates in concentration gradient of nickel sulfate. So let's get started.
The fluidic layer and the control layer are made by replica molding of polydimethylsiloxane, or PDMS, from the SU8 master. To begin this procedure, mix the PDMS prepolymer and crosslinker at a 10:1 weight ratio. Degas the mixture in a desiccator for 1 hour until air bubbles are removed. When the PDMS mixture is ready, place the SU8 master in a petri dish and carefully pour the mixture on top of the SU8 master to the desired thickness. Heat the petri dish containing the PDMS and the SU8 master mold at 80 degrees Celsius for 2 hours to cure the PDMS. After 2 hours, remove the cured PDMS-covered SU8 master from the hot plate and peel the PDMS mold from the SU8 master. The desired structure will be embedded in the PDMS mold. Using a 20-gauge blunt end needle, punch 4 holes in the PDMS mold for tubing to the gradient generator, cell inlet, and cell outlet. Now we are ready to assemble the PDMS device.